tremendous. And I found that to complete one analysis, I need 1,000 hours on my current PC, on this PC. And it turns, it's about 40 days. But fortunately, John loaned me 5 PCs for the Maker Faire. And he hadn't showed up since the Maker Faire. So I brought the computers for him to pick up. He's not there, so I have to take it back. So I got 4 more computers in my house, and I set up a computer farm. I got one computer, this one, and my wife has one upstairs. And I have 4 more computers set up in the lab and have a computer farm. All 6 computers working together on the same piece of RNA. The analysis was done in about 5 days. Okay, so I was very happy to have that part completed. So I show you a picture of my computer farm, and then I will report to you what I really found. And I think I hit the jackpot, okay? Everybody says you're looking for things and you'll find it the last place you look. Yes, this is the last place, okay? So the bioinformatics, generally what the microbiology people, they talk about bioinformation. They're basically talking about the DNA. We have chromosomes. DNAs on all the chromosomes. And then the information on DNA chromosome is transcribed into RNA. Okay. So what they have, our understanding of the DNA mostly is called the coding DNA. That part of the DNA which contains useful information. And the coding DNA get transcribed into RNA. These are the coding RNA. And the coding RNA, what do? They go out and produce protein. So each of this stretch of DNA information in DNA is called a gene. Okay. The old understanding of the DNA or the bioinformatics is that genes on the DNA get transcribed into RNA, and RNA information is used to produce proteins, and that's the general understanding of bioinformatics. Okay. The new bioinformatics, based on my theory, okay, is that you have information on the DNA chromosome, and each cell has a complete information about itself in its chromosomes. The information on DNA chromosomes are transcribed into RNA. But there are 2 different kinds of RNAs. One is called the functional RNA. These are the coding RNA. We understand it. No, we understand them. And mostly they are producing proteins. But I maintain that there are a lot more RNA produced than those which produce protein. And these RNA I call the informational RNA. These carries information. They are not doing things themselves. They are just carrying information which were used to do some other things. And in the functional RNA, I've found lots of RNAs, just long stretch of RNAs. And these are not used to produce protein, and that's what they call the non-coding RNA. And many of these long stretches of RNA, I believe, are functional RNA. They do have biological function. One example is a chromosome RNA, and that's the RNA which builds a chromosome, okay, which is used to produce protein. So in the protein building mechanism, RNA is playing a very important role because it is a structure. This RNA forms a structure, and this structure produces proteins. And there are other RNA, what they call the transfer RNA. And transfer RNA is a small RNA which grabs each different amino acid, and this transfer RNA grabs the amino acid into the ribosome, and then the ribosome uses the messenger RNA build the protein sequence according to the message, according to the information stored in the messenger RNA. So, I believe there are many more RNA which are functional, which serves as catalyst or machinery which has specific functions. But we only know RNA. This is ribosomal RNA. That has important functions. But most of the big RNAs, we don't know what they're doing. But for the informational RNA, these are RNA which contains a lot of microRNAs or information like microRNA. And this is what I call the pairs. Okay, they're small RNA pieces which has the code in it. And this code is not doing anything in themselves, but they have information in it so that it will go out and produce other RNAs. And the other, so this is like a program. We have lists of tokens, okay? And each token would produce another list of tokens. And each token would go out, most likely produce another list. But at the end of this nested list, there are RNAs which are actually functional, which either build protein or do some other useful things. So this is my new bioinformatics. So our understanding of RNA are that there are many different RNAs. Right now we know. So mRNA, these are the messenger RNA. These are used to encode protein, and they are used to build protein molecules. And then tRNA, that's the transfer RNA. I have said that this is in the, while building a complete protein molecule, you need the transfer RNA to bring in individual amino acids and connect these amino acids to form the long protein molecule. So this is a very important. All the cells need them because you need, when you build protein, you need 22 amino acids, and you need at least 22 of the transfer RNAs to facilitate the building. And ribosomal RNA, this is a big molecule which forms the major part major portion of the ribosome. And the ribosome is a mechanism which allows the building of a protein molecule from the messenger RNA. So, but there are a lot more RNAs. Nobody knows what they're doing, okay? But there are lots of them. And so these are called long non-coding RNA because we don't know what they're doing. The old bioinformatics only understands the protein building process. But anything that's not connected to protein building, that's called non-coding. But I believe these are these RNAs do not have code to build protein, but they have code to do other important things, okay? And that's what I want to find out. And then there are microRNAs, very small pieces of microRNA, 18 to 22 base pairs. Lots of them, and many, so far, people have found these are useful things. And they have identified many microRNAs which controls the process of protein building, okay? But I believe that microRNAs are much more important than just control, either accelerate or slow down the protein building process as we currently understand. And there are other RNAs, siRNA, snow RNA, piRNA, sRNA. These mostly people just isolate them from various tissues, and so they give them some strange names. But mostly they don't know what they're doing. Okay, and I want to show you that these microRNAs are actually information carrying stuff, which allows the cell computer to operate. Okay. Any problem? Okay, so my understanding, or from my study of all the RNA, I think RNA can be divided into two categories. The functional RNA. These RNAs do things, and many of them do things very similar to protein. Okay, they facilitate certain chemical reactions inside the cell. So we have messenger RNA, transfer RNA, ribosomal RNA, and other things we don't know. But I believe many of the RNAs have important biological functions. And so these RNAs do things, do important things. But the major part of the RNAs are what I call the informational RNA. These RNAs carry information. They are not doing things by themselves, they just carry information. And what they do mostly is to produce other RNAs. And when you produce a microRNA, and these microRNA are actually instructions in the cell computer, and they either trigger other RNAs to be formed, or they control certain process, certain important process in cells. So in cells, in a cell, there are lots of work has to be done, and there are lots of information which has to be passed along. And I think this is the mechanism how this information are being passed and being utilized to make a living cell. So here I would say that microRNA, I was looking for information just like microRNA, small pieces, but they have carry information which would do important things. And these small stretches of RNA, I would call them pairs. Okay, these are the basic instruction set in a cell computer, and the pairs generally form clusters. They are contiguous clusters of pairs, and this is what I call necklaces. And these are the lists. And the list will perform more complicated tasks according to the microRNA or the necklaces that constitutes the necklaces. So what my work is try to identify pairs, and from the pairs try to identify necklaces. And I believe, although I'm not a microbiologist, so I do not have a lab to carry out work that would identify the functions of these RNAs. But people do silico biology, and that means we use a computer to analyze the information that's inside inside the cell, like DNA and RNA. So I think I can only do work in this area, and hope that will shine some light to the actual microbiologists who can utilize the information here and find what these RNA molecules are actually doing. So I told you that I was, last year I would try to look for the pairs and necklaces in DNA. And I was very disappointed. And so this year I started looking at the long-coding RNA. And because the information on the DNA are generally spread out, so it's not forming recognizable clusters. And the RNA are information which gets transcribed from DNA and gets edited and finally released into the cell to perform certain things. So the information content on the RNA are useful information, okay? So try to find pairs and necklaces in the RNA makes much more sense than try to search this information in DNA itself. So I was looking, there are RNA databases, huge databases, and what I want to concentrate is on the human long non-coding RNA. So these are, I found three interesting libraries, interesting collections of long non-coding RNA. One is LNC pedia version 5.2, and this is probably the most complete collection of RNAs found in human body. And this one, it basically collects all the available information from various sources and deposit them into one huge data file. And this data file for human long non-coding RNA is almost 200 million base pairs. Okay, and this is genome library, human genome library. Human genome library also, has been collecting human genomes for years and years. And so this is the 38, GRCH38. this is human, okay, and then in this collection of human DNA, it has the complete all the chromosomes and all the information, but they have a file which records all the non-coding RNA. But this is only part. It is only 65 million base pair. And I think the NNCpedia include that one. So this one was only in the Human Genome Project have this. And this one has another interesting collection, that's the cDNA. And I'm interested in cDNA because the cDNA are mostly RNA and get reverse transcribed into DNA. And RNA is not stable. DNA is much more stable. So once the RNA gets transcribed, reverse transcribed into cDNA, it can be analyzed and it can be handled much more easily. So there are huge libraries of cDNA available. And this one is a 300 million base pair. So it's a much bigger one. So what I'm trying to do is to write my program, test them on all these 3 files, just to make sure that my tools work properly. Okay, and I wanted to shoot to 300 million pairs, and that's because the largest chromosome in human, the DNA, the chromosome 1, is about 300 million bytes long, and if I can, if my tools can handle this one, it can handle all the genomes. Okay. So most important work is on this NNCpedia. Okay, but the other 2 gives me some way to check on whether I've done things, whether there's any problem in my algorithm. So here, this is my focal point. This collection is collected in Belgium, or warehoused in Belgium, and it has identified 127,000 transcripts, such as stretches of RNA. And there are 56,000 genes identified in them. Okay, and the total length is almost 200 million base pair. So this is my focal point. And then this one, the non-coding RNA and the cDNA, these are collected and warehoused in Cambridge. Okay, so you can download things from various sites, but they mostly have the same information. So I got huge RNA files. How do you proceed to analyze to 300 million bases? Okay, so this is the process of the analysis that I'm doing. So take any RNA file. It is generally the biologists publish their data in what they call the FASTA style. Okay, so there's generally they have a title of a particular gene, and then followed by the data. they are organized so it can be displayed or published on paper. So one line of data probably has 120 bases, and with a carriage return and line feed at the end. So what I do, take any RNA file, they have first line which has a greater sign in front. That's the index line. So I put all the index line in one file, and then consolidate all the data into another data file, and strip out all the carriage return line feeds. Okay, so I just collect all the data in one file, and then have an index file. This index file have pointers to the data file to see to mark where each individual gene is at. Okay, so I only have to save the index and the data file. And data file, because the data are huge, and it's just too much work to analyze each individual file. So I just collect them and make a big flat data file, and then analyze them all at once. Of course, there should be gaps in between genes, but I ignored the gaps. I just analyze them straight through, in hope that when you cross from one gene to another, the data will be discontinuous and would not show up in my per file. Even if they showed up, it doesn't matter. Okay. So in a big data file, I'm looking and extracting all repeated 20 base pairs. Okay, so I just take out 20 bases from the beginning of the file, and ask, is this 20 base pattern repeated? If it is repeated, I'll record both of the location of these two, these matched pattern. And then I move on to the next one, and move on to the next one. And every time I have a repeated pattern, I would record them in my result. Okay. So all repeated, I want to find all the repeated 20 base pair patterns. And these patterns are sorted and packed to variable length pairs. Okay. So I'm looking for 20 base patterns, repeated 20 base patterns. And all these repeated 20 bases are sorted according to the address. And then there are, during the sorting, during the pattern extraction, many repeated patterns will be recorded more than once. Okay, but all the redundant patterns will be eliminated. And then these patterns need to be packed, because when you imagine you have two identical stretch of DNA information, and of course the first 20 base would match, right? So you get that recorded, and you move down to one base down, it still matches because these two are repeated long patterns. So you can see if there are two repeated patterns, you got many of the patterns which are consecutive. Okay, and these consecutive repeated pattern can be consolidated into a long stretch, and that would reduce tremendously the amount of data that's that you originally look up. So, so after if you extract all the repeated base and do the sorting and packing, the data file, the size of the data file is generally reduced by 10, by a factor of 10. And then you got lots of patterns which are variable length, and they are lined up in the data file. So you have a collection of variable length pairs. And once you have identified all the pairs, then you insert the index back. So the index will show up. Okay, here this gene starts here, and then you get pairs, and now next gene starts here, and you get a list of here. So this way you can see the pairs in relation to the genes, okay? And generally, you can see necklaces, which are necklaces in each individual genes in the data file. So this is how data is analyzed. Any problem? Okay. So identifying repeated 20 base patterns is easy say than done. Okay, because when the data file is big, the search time goes to the square of the number of bases. So when you are getting to millions of bases, it will take a long time to do the search. So what I did was to break up the data file into 496 threads, okay? And each thread is related to a 6-base pattern, okay? So 6-base pattern, you have A, C, G, T, A, C, okay? So I'm looking for this pattern in the entire data file, so, and identify each and every occurrence of these 6-base patterns. And this forms a thread, okay? All the patterns that started with this 6-base pattern will have to be in this thread. So I don't have to look anywhere else. I only have to search this thread for a particular 20-base pattern with the same first 6-base pattern. Is it? Is it kind of clear? So this way I can break up the entire data file into 4,096 threads. And for a particular pattern, I only have to search 1/4,000th of the data. Okay, and this will greatly, basically speed up the factor by 4,000 square. Okay. So this is my biggest contribution to this in this work. So the repeated patterns beginning with this particular 6-base pattern can be exhaustively searched in each thread, okay, and the result will be written to a file. So this way, I was generating 4,096 files on my computer. Okay, for each RNA, I generate 4,096 files. And the data in these four files, they are repeated patterns, okay, redundant, okay. This redundant has to be removed. By sort into sequence and then remove the redundant ones. So this way, generally reduce the size of the data file by a factor of by 1/3. Okay, and the totality of this 4,000 file is still too big for the computer to handle. So inside this 4,000 files, I'll take chunks of it, 10 million byte chunk, 10 million base pair chunks, and so each of these 4,000 files, I'll take 1 million base chunk, and then collect all the in the same address range over all these 4,000 files, and collect them into one chunk. And this one chunk are the patterns within this 10 million base range. Okay. So if I collect these chunks, then I can sort them and then pack them again. And this will result in a 10 million base chunk of data and the pairs, a list of all the pairs, okay, all the pairs. And when I have these chunks of 10 million base, the pairs, then I would insert the index file into this 10 million base pair data. And from there, you can identify pairs and necklaces. Okay? 10 million bases. This seems to be the limit of my computer, or the analysis tools seems to fail if I extend it to 2 million byte blocks, and generally I reach I have errors, the memory errors. Okay. So it seems that I can handle 10 million bases quite easily, but anything above that, the computer tends to fail. And for the CDN file, which has 300 million base pair, it took 15 minutes to produce one thread. So I need to produce 4,000 of them, and my calculation is that you need 1,000 hours for 4K thread, and it will take 40 days to do it on my computer, which of course is an old one. It's a 2006 computer running Windows 7. It's the speed, the CPU speed is 2.2 gigahertz. And so I look up the computers. I thought, well, you know, it's almost 15 years. Things must getting much better. So I look up the catalog, and I and then the computer, if it is 10 times faster, I will be I would buy a new one. But But what the advance is mostly in the multiprocessor. So they have built more than one processor into a core. But I don't know how to how to spread the workload onto different processors. Yeah, yeah. Well, so, but so I set up my computer farm. No, I got 2 computers already, and the John didn't take his computer back, so I have 4 more, and I set up 6 computers to work. So in 40 days for 1 computer, when I set up my computer farm, it was complete in 5 days. So that was very, very nice. And I really don't have to go out buy any more computers. So this is the my computer farm with with 2 more computers in other rooms. Okay. Really, you see, the Intel machine are built in search engines. Okay. The Intel processor, 386, 486, 586, they are search engines, okay? Because they have one instruction, the compare P, CMPB, compare bytes. And that's the instruction. If you preceded with a repeat Z, it would repeat the search for whatever the memory size and whatever the pattern you want to search. So it's one instruction, one search instruction that's optimized for search. So did you use a code word then? Yeah. I code the basic compare routine and look routine in the assembly. Okay. So I think. I do the search in Python and other things, and they are 10 to 100 times slower. So fourth is used to do the heavy lifting. Okay, the most basic search work is in fourth, in assembly, and taking advantage of the search instruction in the 386 processor. And once you get the heavy duty processing done, and the other thing can lightweight sorting and packing are all done in Python. And it's much easier to handle files with Python than Forth. And then using once the data are all analyzed, and all sorted and packed, and then the data will be, the final data analysis can be done in Excel. And the the 10 million base pair limit is mostly imposed by Python and Excel, because data any bigger, they seem to fail. And one thing, I was pleasantly surprised that Python was able to open 4,096 files simultaneously for me to pick the chunks. Okay. So the first I thought, well, okay, I got 4,000 files. What am I going to do with them? So I tried to pack them, okay, into a smaller number of files. And I just couldn't imagine that Python can open 4,000 files. So I packed them into 64 files. And so Python has no problem with 64 files. So last week I said, okay, let me just try 4,000 to see what the Python do. And Python has no problem. Open up all 4,096 files, and I just read one chunk from each file and pack them into one chunk file, and then start read the next 10,000 block. So I just keep on doing, and the files are open, so I just continuously reading lines of it until I got to the size I want, and write them out. And this one I can process, 300,000, 200,000, no problem. So I was very pleased to be able to do that. Otherwise, packing and unpacking, it's tedious. But the Python can handle 4,096 file. I think that was amazing. Okay, the results. For this LNCpedia, the final result has nothing but pairs and necklaces, and that was very surprising. And the other files, they produce occasional pairs and necklaces. So you scan through the file, you can find a few clusters of pairs. Long non-coding, so all other files produce occasional pairs and necklace. I think I was happy to see just a few, because it kind of make my point that these pairs need to be connected into necklaces. So just prove the concept that these code are expressed in pairs, and pairs are connected into lists. And so just to support my my thesis that the information in genome are organized in lists, okay, and the nested list can solve any problem in computers, and so it should be able to solve any very complicated problems facing the cell computer. So I said that I hit the jackpot. Okay, so this is the first part of the LNCpedia, and you can see that this repeated pattern very regularly showed up, and it covers from beginning to the end. And it's just like go to Las Vegas, you kind of hit the jackpot and everything. Okay, yeah, they are okay. The first column is the address in the data file, and the next is the distance or the offset from this gene to the next one. So it's the distance between the two genes. And the third one is the length of the gene. Okay.
So each one of those lines are necklaces?
Each of this is a pair. A pair. And all the things, the pairs form clusters, and the cluster is a necklace. Okay. Okay. So let me just show you the data file. Okay. Let's see. Okay, so this is the actual data file. I would just okay, this is at the beginning. So the mark, the index mark, this marks the first gene, okay? And so you can see the next gene. And it just, okay, here are some pairs which are longer, much longer, okay? And so and you get very long, very long. So this one is the 3131 base pair. So that's a very, very long pair, okay? And then the next one is only 177.45, and then there's another one which is 655. So, but mostly they are small pairs, okay? So let me just scan through this thing. This one is what I'm really looking. I'm expecting this kind of pairs, which is about 30 or 30 base, and lots of them, one after the other. Okay. And you can see these things just come along. There are occasionally very long stretches of DNA, RNA, but lots of them are small ones, but occurred one after the other, and that's the nucleuses. So I was first I was got it, I got it, yeah, jackpot. But then I said, well, somehow it looks fishy, okay? Because you see, mostly these are 58. The length, most of this pairs are 58, and they come right one after the other, with only one extra base. Oh, that doesn't look. That looks very artificial. You see here, these are the regular ones that I'm expecting, and here you can see it just occurred repeatedly 58. Okay, and I need a good explanation of that, and I don't have it. So, okay, and you can see that these each gene that's being identified by in the databases. Yeah. Why? Yeah, and I think need to be explained. Mm-hmm. Yeah.
The links,
Mm-hmm. Mm-hmm.
like that.
Yeah. Well.
Okay, and you can come out and get averages and standard deviation and skewness, even the dreaded kurtosis.
Okay.
If you discovered something, you should share it with the world.
Well, I think if the world is ready. Yeah.
So through this whole talk, I was through this whole talk, I was thinking, well, I really want to use the amino acids as gene code words, and
Yes.
I could factor the DNA sequence into 4th words, and then I could run like C to decompile the sequence into an organism, description of the organism, or whatever. And I wonder whether I could do that.
Now, remember, the protein producing the coding part of the DNA is only 1.5% of the entire DNA. So if you concentrate on protein, you are in the wrong track, okay? Because informations are not there, okay?
If I use so is the DNA the source code? Or I guess that would be the source code. I would decompile the protein to the source to the DNA.
Well, this is my decompiler for the human long non-coding RNA. I have decompiled into pairs, and because these are just the genes they have identified and collected. So I really don't want to explain anything between these two genes. Okay, inside this one gene, I think these are connected, but between these are actually different genes collected from different tissues. Okay, so I think the information, the pairs has information in it. Okay, and the nucleus has information in it. Okay, but in between them, they are not connected. Yes. Say it again.
Gene array charts.
No. Okay. This is the. So I'm puzzled. This thing I can explain. These small ones, that these are what I expected, but the 58.
Well, you know what was interesting in that last screen? You know, 58 and 59. 17 is double 58. It's just about double. It's 160.
Yeah. It could be. I don't know. So this is. I worried about it. Okay, because 58. That seems artificial. Okay, and it could be caused by my algorithm or something. And so 58. 58, 61, 58, 59. And this I worried a lot about it. And I thought,
it's a new discovery, though.
Yes, it might be a good discovery, but it also might be something artificial that. Yeah, well, if they are accepted, so I go back to an older analysis. An older analysis, I was just taking 10 million base from the I just divide the data file into 10 million base chunks and analyze this 10 million chunks alone. Okay, so the entire file is 200 million bases. I took only one part, 10 million, and do the analysis.
Can you go to your algorithm again?
Okay. Yeah. Okay.
Ways back.
The,
When we're looking at the data, can you go through the algorithm again?
yeah.
And then could we wrap it up and go to lunch after you answer his question?
Okay. Yeah. Sure. Let me just. I think what I want to. We'll talk about it in lunch. And so this one is an older analysis with only 10 million base chunk. Okay, and you can see the result is what I generally expected. Okay, but when you enlarge the analysis to cover the entire 200 million base, then the pairs seem to fill up each individual. Let me just show you the beginning of it. You can see if I analyze only the first 10 million base chunk, I got 5 pairs, okay? But if I enlarge the whole thing into the entire 200 million base, okay, here you see these are filled in. You see, this is only the first 10 million base chunk. I got only 5. But when I analyze the whole thing, I got a whole bunch of these 58 base pair patterns. So I think these are real, okay? Because if I only analyze a portion of it, this is the same analysis algorithm. It's just that this one is analyzing 200 million bases, and this one only analyzes the first 10 million bases. So I think the algorithm is okay, okay? And it is data. When I think the LNCpedia collection is probably the complete collection, okay? And so it shows me all the pairs. No, very few, you see, very few. Yeah, very few, okay? But when you enlarge the analysis, things get filled in, and the gaps.
So going back to here, the
Yes.
first one is the offset into
This is the address of the huge data file. The address
the address of
in bytes. It's just a whole you see the entire collection has many genes.
Right, but I mean, that's each one is a block, isn't it? It's not a distinct address. It's small.
This is the address. We are at the very beginning of the entire data file.
The file address?
Yes, the byte address, okay? So byte address, and this is the offset, this gene to the distance from this gene to the next gene, and this one is the actual length of this gene, okay? All right, okay, I think that's almost everything I have here. Let me just go back to the to this thing. Okay. Computer form analysis, and the results. This one. Okay. So I've shown you the scan file. And so here, lots of consecutive 60 by 60 base pair pairs, and hence necklace. Lots of necklace with 30 base pair pairs. But there are also very long stretches of the RNA. And these long stretches are actually repeated in the database. So these are interesting things, and may have lots of meaning, but I'm only a programmer, okay? I'm not a biologist. So these need some expert to do the explanation. And are these 60 base pairs real? Okay. Are they real? Do they have actually meaning, biological meaning? I don't know. And the necklaces are too good to be true. Okay, so each gene that's being identified are now represented by a necklace. Is that coincidence? Or does that has a real physical or biological means? These wall-to-wall necklaces are not present in the in this smaller file, okay, or in the cDNA. Why? I don't know. Yeah. So just one moment.
Last one, cDNA file is just most of the transcripts are transcribed into protein. So maybe the protein encoding transcripts don't have such pairs and like this.
These are non-coding RNA.
cDNA one should.
cDNA is also non-coding. Okay.
So how many pairs you can find in one necklace? Or how many in each?
Well, I haven't got all the statistics yet, but I think you can see there are many. So these are generally in the tens of thousands. Okay.
How many these pairs in one?
Yeah, that depends. All the genes are of different size. So there are lots of genes which have thousands and thousands of pairs. Okay. Mm-hmm. So these are all varied, and that's good because if they are all the same, definitely I've screwed up. Okay. But I think I'm doing the algorithm-wise. I think they are good. Okay. They are reliable, but biologically, I don't know. You guys have to figure it out. Okay, yeah, thank you very much. Okay, we'll go to lunch, and we'll be back on the air on 1 o'clock or 1:30. Okay, yeah. Thank you. Yeah. Yeah.
Each generates you because each computer